Good morning, good afternoon, and good evening, everyone. Welcome to our global webinar series hosted by Elabscience Bioinnovation. We are pleased to have participants from around the world joining us today for this advanced session on Cell Death Atlas, a holistic view of cell death pathways. Let me introduce myself first. I am Dr. Ikra Hafiz from Elabscience and I am the member of Global Scientific Affair Department and I will be your host for today's session where we will delve into the intricate landscape of regulated cell death and its fundamental significance in biological system disease mechanism and therapeutic research. Let's begin this journey into cellular fate and scientific discovery. A digital participation certification will be provided to those who attend the live session and the session recording will be sent to all registrants. Attendees also have the chance to receive free sample of selected Elabscience product. Highlights are on the slides and the full list with eligibility and claim instruction will be sent to you by your registration email. Now it's my pleasure to introduce today's speakers, Dr. Sundas Rana Qureshi, who will guide us through today's session on Cell Death Atlas, a holistic view of cell death pathways. As we explore this theme, we will gain insight into the diverse mechanism of regulated cell death and how they shape key process in health, disease progression, and therapeutic research. We are excited to learn from her insights today. And Dr. Sundas, over to you.
Okay, so welcome everyone to today's session on Cell Death Atlas, a holistic view of cell death pathways. Actually, cell death is not a new phenomenon, but during this studies, during more and more research, new phenomena do occur. And as the time passes, we are getting to know more about cell death, more pathways, and how does they work. So that's why we decided to give you a holistic view of these pathways so that you can use them in your research. So let's begin properly. Okay, most of the people hear the term cell death and immediately think of loss, but in biology, death is often essential for life. During embryonic development, for example, apoptosis helps separate out fingers and toes by removing unnecessary cells, like, for example, in the tadpole development into the frog. But in healthy adult tissues, old or damaged cells are constantly removed and replaced to maintain that balance. Your immune system also relies heavily on cell death to eliminate infected or potentially dangerous cells before they become a problem. But when these death programs go wrong, biology starts to unravel. Too little cell death can allow cancer cells to survive for longer period of time than they should. Too much cell death, on the other hand, contributes to neurodegeneration, organ injury, and chronic inflammation. This is also why cell death has become such an important therapeutic target. Many modern drugs are essentially designed to either trigger the death or in some harmful cells, or sometimes to prevent death in vulnerable ones. Traditionally, when people talked about cell death, they mostly meant apoptosis. For many years, that was the dominant framework. But biology turned out to be much more complicated than that. Cells don't die through just one program. They have multiple death routes, each triggered by different stresses and producing different consequences. Apoptosis is classical programmed death pathway. Everybody knows that. Clean and usually non-inflammatory. Autophagy is a bit different. It often helps cells survive under stress, but under certain conditions. Excessive autophagy also contributes to cell death. Then we have another pathway, which is necrosis, This is old textbook example of accidental death caused by severe injury or toxicity. Then later on, we discovered regulatory necrotic pathways like necroptosis, where cells actively execute a necrosis-like program. Pyroptosis is highly inflammatory and plays a major role in infection and innate immunity. Then we have the ferroptosis. This brought completely new concept, death driven by iron-dependent lipid peroxidation. That changed how we think about oxidative damage. More recently, Cuproptosis and Disulfidptosis also show us that metal stress and disulfide stress can also drive unique death mechanisms. And then there is PANoptosis, which is especially interesting because sometimes dying cell doesn't follow a single pathway at all. Instead, it activates a coordinated hybrid program involving apoptosis, pyroptosis, and necroptosis simultaneously. So rather than thinking of these as isolated boxes, it is more accurate to think of cell death as a connected network that constantly crosstalk. Now, the obvious question is why would cells need so many different ways to die? They can die just by one process. Why isn't apoptosis enough? The short answer is that not all cellular stress is same. A cell dealing with DNA damage is facing very different program from a cell infected by a virus or overwhelmed by oxidative stress. The cell has to interpret what kind of damage it is experiencing and how severe that damage is. Sometimes the safest option is a clean, controlled removal through apoptosis, where neighboring tissue is barely disturbed. But in case of infection, silence can actually be dangerous. So, if the pathogen is replacing or replicating inside the cell, the immune system may benefit from an inflammatory death like pyroptosis that essentially screams for help. That is why the outcome matters just as much as the trigger. And this becomes very important experimentally because two dying cells may look similar under the microscope while following completely different death programs. It is very important to know the difference between classical death pathways and the new regulated ones. So let's discuss what classical death pathways we know. The first and foremost one is the apoptosis, which is the most classical form of cell death program, and probably the most studied one as well. It's highly controlled and energy dependent. There are two major ways that apoptosis can begin. The first is the extrinsic signaling, where external death ligands such as Fast ligand or TNF bind to receptors on the cell surface. That activates the caspase 8 and initiates the death program from outside the cell. The second route is intrinsic signaling, which responds to the internal stress like DNA damage, oxidative stress, or even hypoxia. This pathway resolves around the mitochondria, which acts almost like the cell's final checkpoint. The balance between pro survival proteins like BCL2 and pro death proteins like BAX and BAC determine whether the cell commits to the death. Once the mitochondrial membrane becomes permeable, cytochrome C is released into the cytoplasm. This leads apoptosome formation and activation of caspase 9. Whether the signaling starts externally or internally, both routes eventually cover or based on the caspase 3 and caspase 7, which carry out the final demolition of the cell. When studying apoptosis, relying only on morphology is usually not enough because several death pathways can look similar in the early stages. That is why we use biomarker panels rather than single readouts. Caspase 3 and Caspase 7 are among the most trusted markers because they represent the execution phase of apoptosis. PARP cleavage gives additional information that caspases are actively dismantling the cell. Annexin 5 with PI, propedium iodide. This is essentially useful when you want to distinguish early apoptotic cells from late apoptotic or even sometimes necrotic populations. For the DNA fragmentation, tunnel assay remains a widely used approach, especially in the tissue sections. And backs to BCL2 balance often tells you whether a cell is being pushed towards the survival or towards apoptosis. Now, the next most important cell death, or you can say classical cell death pathway, is the autophagy. This is quite different from apoptosis because it isn't always a death pathway. It is strange, but it is true. In many situations, it acts like a survival mechanism. When cells face stress, they stresses like starvation or damaged organelles. They need a way to recycle internal components and then generate energy in the form of ATP. That's where autophagy comes in. The cell starts forming a double membrane structure called an autophagosome, which surrounds the unwanted cargo like protein aggregates or dysfunctional mitochondria. That vesicle then fuses with the lysosome where the contents are degraded. The breakdown products can be reused for biosynthesis or energy production. You can think of autophagy as a cell recycling system because this will be very easy to remember. But like many biological processes, too much of the good thing becomes harmful. If autophagy becomes excessive or uncontrolled, the cell may essentially digest itself beyond recovery because this is actually a digestion mechanism. Okay, for autophagy, biomarker interpretation can be a little tricky because increased marker levels do not always mean increased autophagic activity as well. That is why LC3 conversion comes in from LC3-1 to LC3-2, which is one of the most widely used readouts for autophagosome formation. Then we have another marker like P62. It is also very informative because it gets degraded during the active autophagy. So if P62 accumulates, it may usually suggest impaired flux rather than increased degradation. Another marker, which is Bacillin-1, that helps us assess early pathway initiation. ATG5 and ATG7, these are also key components of autophagic machinery and are commonly monitored in mechanistic studies. Now, the necrosis, really difficult one. This is probably the most familiar form of cell death as well because it happens during severe injury. You get hit and necrosis happens. Unlike apoptosis, necrosis is not a controlled self-destruction program. It usually occurs when the damage is simply too overwhelming. You commonly see this in trauma, toxin exposure, or ischemic injury where ATP level collapse. The cell first starts swelling up because ion balance is lost. Organelles enlarge, membrane integrity weakens, and eventually the plasma membrane ruptures. Once that happens, intracellular components spill into the surrounding tissue and ultimately trigger the inflammation. So it is very easy to find out a necrosis happening. For necrosis, the most informative biomarkers usually reflect membrane damage because membrane rupture is the defining event. LDH release is widely used in cytotoxic assays since LDH leaks out once in the membrane integrity is lost. PI or propydium iodide is another classical marker because it can only enter the cells when plasma membrane becomes permeable. HMGB1 is particularly interesting when it acts as a danger signal after being released from the damaged cells. That release helps explain why necrosis is often strongly associated with the inflammation and tissue injury. Now that we have covered the classical ones, there are many, many new emerging regulated cell deaths. So let's have a look on them. The first one, really, really similar to necrosis, which is necroptosis. This is a fascinating pathway because it sits right between apoptosis and necrosis. Morphologically, it resembles necrosis. The cell swells, the membrane breaks, and intracellular contents leak out. But mechanistically, this is not accidental damage. It is a tightly regulated process. It usually starts with extracellular death signals, most commonly through TNF receptor activation, whether it's alpha or beta. Under modern normal conditions, those signals may lead to apoptosis, especially if caspase 8 is active. Caspase 8 essentially acts like a gatekeeper here. As long as it is functioning, necroptosis is usually suppressed. But when caspase 8 is inhibited, something that can happen during a certain viral infection or inflammatory condition, cell activates a backup death pathway. That is when RIPK1 and RIPK3 interact and resemble into a complex called necrosome. This complex drives a phosphorylation of MLKL, which is a key executioner in necroptosis. Activated MLKL then oligomerizes and translocates to the plasma membrane. Once it reaches the membrane, it disrupts the membrane integrity and forms destructive pores. The cell eventually ruptures, releasing DAMPs and triggering strong inflammation. In simple terms, necroptosis is what happens when apoptosis is blocked, but still needs to die. When we are studying necroptosis, the most reliable biomarkers are centered around RIPK3, MLKL signaling axis. Phosphorylated RIPK3 is one of the earliest indicators that necroptotic machinery has been activated. Once RIPK3 is active, it phosphorylates MLKL. So PMLKL is often considered one of the strongest markers for pathway confirmation. I'm talking about phosphorylated MLKL. Then the MLKL oligomerization is even more informative because that step directly links the signaling to membrane disruption. Another important clue is caspase 8 inhibition. That's particularly useful when distinguishing necroptosis from apoptosis is essential because in apoptosis, caspase 8 is active, while here its suppression often enables the pathway. In practice, researchers usually combine at least two of these markers for the confident validation. Now, the next one is pyroptosis. Also very interesting because of its most inflammatory form of cell death. And it plays a very major role in innate immunity. Unlike apoptosis, where cells die quietly, pyroptosis is designed to alert the immune system as aggressively as possible. It usually begins when the cell senses the danger signals such as bacterial toxins, viral infections, or intracellular stress. These signals trigger assembly of inflammasomes, which are larger protein complexes acting like intracellular danger sensors. Once formed, the inflammasomes activate caspase 1 in the canonical pathway. There's also non-canonical pathway involving caspase 4, caspase 5, or sometimes caspase 11, depending upon the species. These caspase cleaves actually gasdermin D, which is the key executioner of pyroptosis. The end terminal of the fragment of gasdermin then inserts into the plasma membrane and forms the pores. These pores completely change the membrane permeability because water rushes in and the cell swells and eventually ruptures. At the same time, inflammatory cytokines like IL-1 beta and IL-18 are also released into the extracellular space. That cytokine release acts like a distress signal to the nearby immune cells. This is why pyroptosis is heavily studied in infection, sepsis, and hyperinflammatory studies. Okay, when we are validating pyroptosis, the strongest biomarker usually reflects both pore formation and inflammatory signaling. So, the cleaved gasdermin D, especially the N-terminal fragment, is one of the most important markers because it directly forms the membrane pores. Caspase 1 activation is another classic readout for canonical pyroptosis. If you suspect a non-canonical pathway, you may also look at caspase 4, caspase 5, or caspase 11, depending upon your model system. IL-1 beta and IL-18 are especially valuable because pyroptosis is not just about the cell death, it is also about the cytokine release. That inflammation output is really what makes this pathway biologically powerful. PI positivity is also commonly used because gasdermin pores increase the membrane permeability enough for PI to enter the cell. In most studies, combining gasdermin cleavage with cytokine measurement gives strong evidence for pyroptosis. Now, the relatively newer form of regulated cell death is ferroptosis, and it is fundamentally different from apoptosis or necroptosis. The central driver here is iron dependent oxidative stress. So, it is entirely different from other forms which we have studied until now. When the intracellular iron level increases, especially ferrocine, it can participate in the reactions that generate highly reactive oxygen species. The ROS molecules then target the polyunsaturated fatty acids in the cell membrane. That leads to lipid peroxidation, which is essentially oxidative damage spreading along the membrane structure. Unlike the enzymatic signaling pathway, this process is more chemical and catalytic in nature. Once lipid damage starts, it tends to propagate, creating a chain reaction across the membrane. Mitochondrial function is also affected, but the key event is really the membrane level oxidative breakdown. In many studies, GPX4 acts as a protective enzyme because it reduces lipid peroxide. So when GPX4 is inhibited or depleted, cells become highly vulnerable to ferroptosis. This mechanism is particularly important in the cancer biology, where iron metabolism is often dysregulated. It is also being actively studied in kidney injury and neurodegenerative diseases where oxidative stress plays a major role. Because of its unique mechanism, ferroptosis is now considered a promising therapeutic target in multiple disease contexts. Okay, now experimentally looking at the ferroptosis. We usually rely on the combination of biochemical and metabolic markers. One of the key regulators we just discussed is GPX4, which normally protects the cell by reducing lipid peroxides. And when GPX4 is inhibited or functionally lost, cells become highly sensitive to ferroptotic damage. Another strong readout is lipid ROS accumulation, often measured using probes like C11 body P This reflects the actual oxidative damage occurring in the membrane lipids. ACLS4 is also important because it increases the susceptibility of membranes to peroxidation. And finally, the most important one, iron dependency, which is the defining feature. Since chelators like DFO can block the process, so it is really important to check the iron dependency. When these markers align together, it gives a very strong indication that ferroptosis is occurring. Metal is just in the form of metals. Not only iron is making this change, making cells to die. There are some other culprits as well, like copper. So, Cuproptosis is a relatively newly characterized form of cell death, and it is quite different from the oxidative or inflammatory pathways we have discussed earlier. Here is the central player, which is copper, specifically intracellular copper accumulation. Under normal conditions, copper is tightly regulated because it is essential for enzyme activity. But in excess, it becomes toxic. What makes cuproptosis unique is its direct connection to the mitochondrial metabolism. It specifically targets the cells that rely heavily on TCA cycle and oxidative phosphorylation. Inside the mitochondria, certain enzymes are lipolylated, meaning they carry lipoic acid modifications which are required for the metabolic activity. When copper binds to these lipolylated components, it leads to abnormal aggregation of the proteins such as DLAT and DLST. This disrupts the normal flow of TCA cycle. At the same time, mitochondrial structures begin to collapse, especially the cristae organization. Another marker, which is FDX1, this has been identified as important regulator in this process because it influences the copper reduction and sensitivity. As the damage progresses, what happens? ATP production declines and metabolic balance is lost. Eventually, the cell undergoes a form of metabolic catastrophe rather than a classic apoptotic signaling. Because of this mechanism, cuproptosis is being actively studied in cancer metabolism and mitochondrial disease context. And it provides a completely different angle on how metal ion hemostasis can control the cell fate. In cuproptosis research, we usually focus on a very small set of mitochondrial and copper-related markers that reflect the core mechanism. One of the earliest signs of copper accumulation, especially can be revealed when elevated copper ion levels can be seen inside the cell. This is often measured using specific copper detection assays or mass spectrophotometry based methods. FDX1 is important marker I just mentioned because it regulates how cell process and respond to copper. Downstream, we can also see the aggregation of lipolylated mitochondrial proteins like DLAT, which is very characteristic event in this pathway. This aggregation disrupts the normal enzyme function inside the TCA cycle. And at the same time, mitochondrial respiratory chain activity starts to decline due to the structural and enzymatic disruption. Another supporting feature is the loss of Fe-S clusters, which further destabilize the mitochondrial membrane. When these markers are seen together, they strongly point toward the copper-induced metabolic toxicity rather than other forms of cell death. Now, another most important form of death is when a disulfide collapse. This is also one of the new and less familiar forms of regulated cell death, so it needs a bit more context to understand. It starts with the imbalance in cysteine uptake, often through the transport systems like SLC7A11, where the cell brings in more cysteine than it can efficiently process. To cope with that, the cell starts consuming large amounts of NADPH, which is normally essential for maintaining the redox balance. As this continues, NADPH level drops and the cell gradually loses its reducing capacity. At that point, disulfide bonds begin to form abnormally within the intracellular proteins. This shifts the problem from a metabolic imbalance to a structural one. Protein links to the cytoskeleton, they are also especially affected. And then actin filaments begin to lose their organization. As the actin structure collapses, the overall shape of the cell and the mechanical stability start to fail. Unlike apoptosis or ferroptosis, this pathway does not depend on caspase activation or lipid peroxidation. Instead, it is driven by redox exhaustion and abnormal protein bonding under stressful conditions. Because it is relatively recent, the exact disease connections are still being mapped. Current evidence suggests that it becomes more relevant in metabolic stress situations where cysteine handling is disrupted. Disulfidptosis is closely related to cysteine transport, we just studied, and also the intracellular redox balance. Now here, the key feature is overexpression of SLC7A11, which increases cysteine uptake into the cell. When too much cysteine enters, the cell tries to compensate by consuming large amount of NADPH. Over time, this leads to depletion of cell reducing power. Once that balance is lost, disulfide bonds begin forming abnormally within the structural proteins. The actin cytoskeleton is particularly sensitive to this change and start to lose its organization. This structural collapse is what ultimately drives the loss of cellular integrity in this pathway. Now, another very interesting concept, which is PANoptosis, because it brings together three major regulated cell death pathways into a single integrated framework. Instead of thinking about apoptosis, pyroptosis, necroptosis as separate endpoints, this model suggests they can interact and even activate together under certain conditions. At the center of this system is the multi-protein complex called panoptosome. This structure actually acts as a signaling hub where different death pathways can converge depending upon the type of stimulus the cell receives. For example, the components from apoptotic signaling, such as caspase-related proteins, can coexist with pyroptotic elements like gasdermin activation. At the same time, necrotic machinery involving RIPK1 and RIPK3 can also be engaged. What makes this important is that in many biological settings, especially during the infection, cells do not follow a single clean death pathway. Cells are weird. So instead, what they do, they activate overlapping mechanism depending upon the pathogen type, immune signaling, and intracellular stress levels. This is particularly relevant in host pathogen interactions where viruses and bacteria often trigger mixed responses rather than isolated pathways. It also helps explain why in some experimental systems, blocking one pathway does not fully prevent the cell death. So rather than a linear decision tree, PANoptosis represents a more networked and more flexible system of cellular demise. When we move from concept to detection, PANoptosis is usually studied through a combination of key biomarkers from all three major pathways. At the center is panoptosome complex, which includes the proteins like ZBP1, RIPK1, and caspase-8. This complex acts as the signaling hub that coordinates multiple cell death programs at once. To confirm apoptosis involvement, we typically look at caspase-3 activation. For pyroptosis, GSDMD cleavage is a key indicator of membrane pore formation. For necroptosis, MLKL phosphorylation is commonly used to track membrane rupture events. So when these markers appear together in the same system, it suggests a coordinated multiple pathway activation rather than a single isolated death program. Now we have already discussed almost all the death pathways which we know till date, but there are specific detection and strategies for these. So let's see what are those. When we study cell death, no single method is enough to capture a full picture, what's happening inside the cell. Different techniques capture different layers of same biological process, and that's why we always need a combined strategy. Morphology is usually the starting point where we observe visible structural changes such as cell shrinkage, swelling, or membrane bulbing, specifically in the apoptosis. It gives a quick overview, but it cannot explain the underlying mechanism. Fluorescent-based assays move us a little closer to molecular events, allowing us to track signals like ROS production, membrane integrity, or caspase activation. Then we have the biochemical assays. They quantify what is happening inside the cell, such as enzyme release, ATP depletion, or metabolic stress levels. We also have protein-based methods like Western blot or ELISA. They help confirm specific pathway activation by detecting key biomarkers. Live imaging adds a dynamic perspective, showing how cells progress through different stages of the cell death over time. This is especially useful when pathways overlap or when timing of the event is critical. In real experiments, these methods are not used in isolation but combined to reduce misinterpretation. That integrated approach is what allows us to confidently distinguish between different cell death mechanisms in complex biological systems. Okay, when we are designing the experiments for cell studies, the first step is always to define what exactly you are trying to confirm. Different biological questions require different types of readouts, so the choice of assay is not random. If we are looking at early pathway activation, flow cytometry, and ROS-based biochemical assays are often more informative. When we focus on the secreted or extracellular signals, ELISA becomes more relevant. For confirming the pathways like specific proteins, Western blot is usually the most reliable approach. Immunofluorescence helps us understand where these changes are happening inside the cell, which adds the spatial context. If we need to observe the structural or morphological changes, imaging-based approaches give a broader overview. So in many cases, one assay is not enough, especially when pathways overlap or show similar markers. That's why combining techniques is important to reduce the interpretation bias. The key idea is to match the assay with the biological question rather than forcing a single method for all situations. This approach helps ensure that the data we generate is both specific and biologically meaningful. Now, in a real experimental workflow, the difficulty is rarely identifying whether a cell is dying, but figuring out which pathway is actually responsible. So several forms of death share overlapping features, which is why interpretation can easily become misleading if one marker is used. Apoptosis is usually regulated and maintain membrane integrity until the late stages, which is one of the key distinguishing features. Necrosis behaves differently, usually resulting from severe injury and showing early loss of membrane. This membrane integrity can also be without any regulatory control. Necroptosis. This sits between, since it is programmed but leads to membrane rupture. So, through a distinct signaling route, it can be detected. Pyroptosis is another regulated pathway, but it is strongly inflammatory and often linked with the infection related signaling. Morphological changes alone can be deceptive because swelling or membrane disruption can appear in more than one pathway. This is where molecular markers become essential for clarity. Like, for example, caspase 3 activity is strongly associated with apoptotic execution. MLKL phosphorylation points towards a necrosis-driven membrane disruption. Gasdermin D cleavage is key feature of pyroptosis pore formation. So even commonly used readouts like PI uptake or annexin 5 binding can also give you a contextual interpretation since they are not pathway specific on their own. Reliable identification only comes from combining morphology, biochemical signals, and pathway specific proteins together. Without that integrated approach, it becomes easy to misclassify overlapping cell death phenotypes in experimental systems. So, I hope that this lecture today has given you a sense of how you can detect different forms of cell death. So, in the cell death research, one of the most common issue is not generating data but interpreting it closely. A major challenge is the false positives, where signal appears strong but does not actually represent true pathway activation. This often happens when assays are sensitive but not specific enough for a single mechanism. Another problem is biomarker overlap, where different cell death pathways share the same molecular indicators. In reality, most of the pathways require multiple layers of confirmation before any conclusion can be made. So, assay interpretation also plays a key role, especially when the experimental context is not considered carefully. Even technically correct data can lead to incorrect biological conclusions if the context is missing. That is why relying on isolated readouts is not sufficient in the complex systems like regulated cell death. A proper experimental strategy always requires cross validation across different methods and markers. Without that, you will be risking misclassifying the actual biological process, which becomes significantly high. So that's all for today. And I hope you got an idea of what we can help you here at Elabscience. I will be taking questions. We don't have so much time, but you can write the questions in your chat box. And right now, I would just simply give the floor to Dr. Ikra.
And first of all, I want to say thank you, Dr. Sundars, for that excellent overview of the diverse cell death pathway and their biological significance. As our understanding of regulated cell death continues to evolve beyond the traditional concept of apoptosis and necrosis, researchers increasingly require integrated approaches to investigate multiple cell death mechanisms simultaneously. The Cell Death Atlas provides a comprehensive framework for exploring major cell death modalities, including apoptosis, autophagy, necrosis, necroptosis, and pyroptosis. By highlighting key molecular biomarkers such as caspase, LC3, P62, RIPK3, MLKL, and Cas-termin D, it helps researchers navigate the complex network of cell death pathways and their biological interaction. This integrated resource enables scientists to identify relevant biomarkers, understand underlying molecular mechanisms, investigate pathways, crosstalks, and accelerate discoveries in disease research, biomarker development, and translational application. Let me now walk you through the novel cell death pathways and Elabscience detection solutions. As we know, Ferroptosis, Cuproptosis, and Disulfidptosis and PANoptosis are emerging regulated cell death pathways that highlight how metabolic imbalance and organelles dysfunction determine cellular fate. First, we will focus on ferroptosis and cuproptosis, which are two distinct forms of cell death driven by iron and copper metabolism disturbances. Ferroptosis is characterized by iron dependent lipid peroxidations marked by lipid ROS accumulation, uh, glutaperoxidase 4 inhibition, ACSL4 upregulation, and iron overload. While cuproptosis is triggered by mitochondrial copper accumulations, leading to proteotoxic stress, disruption of lipolytic TCA cycles, proteins such as DLAT and DLST and FTX1 dependent metabolic dysregulations that impairs mitochondrial function. To support the investigation of these mechanisms, Elabscience provides a comprehensive portfolio of detection solutions enabling precise assessment of oxidative stress, metal ion imbalance, lipid peroxidation, and mitochondrial dysfunction, thereby facilitating reliable and in-depth analysis of ferroptosis and cuproptosis pathway in biomedical research. Secondly, we will discuss disulfidptosis and PANoptosis. Cell death pathways expanding cellular fate beyond classical mechanisms. Disulfidptosis is driven by disulfide stress from cysteine overload, leading to redox imbalance with NADPH depletion and abnormal actinic disulfide cross-linking. While PANoptosis is an integrated inflammatory cell death pathways combining apoptosis, pyroptosis, and necroptosis, characterized by caspase-3 activation, GS-DMD cleavage, and MLKL phosphorylations. Further, to support these studies and your research work, Elabscience are always here. Elabscience provides a key assays including NADPH, NADP, and GSH, GSSG for disulfidoptosis and caspase-3, caspase-1, interleukin 1 beta, interleukin 18, TNF-A, RIPK3, and MLKL for comprehensive PANoptosis analysis. Now we will move on the question and answer segment. Please type your question in the chat box and we will address as many as possible. If we are unable to answer all questions during this time, you may also reach us at event@elabscience.com for detailed follow-up responses. I have a quick question. So the question is, I am designing a cell death experiment from scratch, but I keep getting stuck at the very first step. How do I actually decide what pathways to even focus on without overcomplicating it from the beginning? Dr. Sundas, can you answer this?
Okay, in practice, the starting point is always the biological context of your model rather than the pathways themselves. You first look at what kind of stress or what kind of trigger your system is exposed to. For example, chemical toxicity, oxidative stress, infection, nutrition deprivation, or receptor stimulation. Each of these trends to bias the system towards a certain death program, even if multiple pathways are possible at the same time. Once the context is clear, you can narrow down to a small set of likely candidates instead of trying to cover all 9 pathways in parallel. From there, you build a minimal marker panel that can separate those possibilities early. The key idea is to avoid full coverage thinking at the beginning and instead move in layers so that you can get a broad hypothesis, then narrowing down based on your data.
So let's take another question, and other question is, in many of my experiment I see markers for apoptosis, necrosis and sometimes even inflammatory signals at the same time. How should I interpret this kind of mixed readout without assuming I have done something wrong? And Dr. Sundas, will you take this question? Okay.
Okay, mixed signals are actually very common in real biological systems, especially when the stress level is strong or it is prolonged. Cells rarely behave like isolated textbook pathways. Instead, they often show overlapping activation depending upon timing or sometimes severity. For example, apoptosis can start in a regulated way but later progress into the secondary necrosis if clearance doesn't occur. At the same time, stress signals can activate the inflammatory components in parallel. So instead of thinking something is wrong, it is better to interpret it as either temporal progression or pathway crosstalk. This becomes most important missing piece in these situations where usually time is the resolution. A single endpoint snapshot often compresses the multiple biological stages into one confusing readout.
So let's take another question. The question is, I ran an Excel 5 and PI staining, and now I have multiple populations showing up, some annexin positive only, some double positive and some PI only. But I am unsure how to confidently translate this into a biological conclusion. Dr. Sundas.
Okay, let me read the question again. Uh, Someone is asking, I ran an XN5 and PI staining and now I have multiple populations showing up. Some NXN positive only, some double positive and some PI only. Okay. Actually this pattern is expected in dynamic cell death systems and each population reflects a different stage rather than a different pathway automatically. An XN5 positive with PI negative generally reflects the early membrane changes before full loss of integrity. Double positive cells are usually in late or transitional stage where membrane disruption has already started. You also asked about PI only population. This often indicates that cells have already lost the membrane integrity without any clear apoptotic progression. The most important point here is that these categories describe stages of cell status, not definitive pathway. To convert this into a pathway level conclusion, you need to use at least one intracellular marker such as caspase activation or MLKL signaling combined with the morphology or biochemical validation.
So we have less time and we have to cover all the question quickly. Next question is, I often see strong caspase 3 activation in my system and I am tempted to directly label the process as apoptosis. But I have also heard that this can be misleading, and how strict should I be with that interpretation? Dr. Sandas?
Okay. You're asking about caspase 3 activation. Actually, caspase 3 is indeed a central executioner in apoptosis. So, strong activation is a meaningful signal. However, it should be treated as a major component of apoptosis rather than a standalone definition of it. There are some situations where caspase activation occurs without full execution of classical apoptotic morphology, or where the downstream processes diverge depending upon the cellular context. For a confident conclusion, you usually want to align between the caspase activity, PARP cleavage, and morphological features such as cell shrinkage and membrane bulbing. Without that alignment, caspase alone is suggestive but not fully definitive. Dr. Ikra, I think the time is finished, so we have to wrap up.
Okay. So the time for today's webinar has now come to an end, and thank you all for joining us and actively participating in this session on Cell Death Atlas, a holistic view of cell death pathways. We hope Dr. Sundar's presentation provided valuable insight into the mechanism of regulated cell death and their significance in understanding disease biology and advancing life science research. Here is reminder, if any question remain unanswered, please feel free to contact us at elabscience.com, and our team will be happy to provide detailed follow-up support. And please also check your registration email for information on accessing the session according and related resources. We look forward to welcome you to our future webinars. Have a great day and happy experimenting. Okay. Bye bye.